Hi y'all. I am continuing talking about the chart, a little bit more about biochemistry, so we'll go into proteins in a little bit more depth, and we're going to talk about dehydration synthesis and hydrolysis in very broad terms. So let's get right back to the review. When we're talking about proteins, it's not just enough to talk about, oh, well, the polymers are called polypeptides. We also think about the way these polypeptides are structured. And there's four basic categories of protein structuring, and they each build upon the first. So we'll start with primary structure. Let me see if I can't bring this just a little bit closer so you can see a little bit better. Although all of these will be posted as individual pictures for your use as well. Primary structure is where we have amino acids combined together using a peptide bond. That is the simplest structure. This is what we get when we go through translation. Secondary structure has that primary structure, but it's going to fold or twirl them because we have these hydrogen bonds between individual amino acids. So remember that if we're talking about a curl, that's an alpha helix. If we're talking about a pleat like this, so just like your skirts are pleated, it's called a beta pleated sheet. Tertiary is where we have a combination of primary structures and secondary structures that are just continue to have specific bonds in between them, to add a little bit. So these have the peptide bonds, which are primary. They have the hydrogen bonds, which are secondary. And then they also have more hydrogen bonds and something called a disulfide bridge, which is a bond between two sulfide structures. In addition, this is where the composition of the amino acids matters, because we're thinking about things like, is it polar or nonpolar? Polar stuff stays together, nonpolar stuff stays together. Is it positively or negatively charged? What are some other things? Polar, nonpolar, positive, negative, acid or base. So all of these things are going to cause some internal folding that's going to give more structure to the shape. Quaternary is where we have two or more tertiary proteins combined. So something like hemoglobin actually has four individual tertiary components that are bound together. Enzymes would be a great example of a tertiary or possibly a quaternary structure, and it's one of those really specific types of proteins that we would definitely want to just kind of review. The cool thing about enzymes is their specificity. So we have our little enzyme structure right here. The little green thing represents a substrate, which is the thing that it's going to work upon. And then we have inhibitors. There's an inhibitor that can go into the active site, just like the substrate can. So that is a competitive inhibitor. And then we have an inhibitor that can go somewhere else on the three-dimensional enzyme shape to inhibit its function, and that would be a non-competitive, some people call it an allosteric inhibitor. If the inhibitor is inside the enzyme, it's not going to be able to function as long as the inhibitor is there. It basically bends it out of shape in a way that the substrate will not be able to get in to do what the substrate's going to do, or to have done to it, rather. If there's no inhibitor in place, the substrate can fit inside the active site right here, which is where the chemical reaction occurs. And when we look at the whole thing combined, we call that the enzyme-substrate complex. This is an example of modeling in biology. Now, when we talk about modeling, remember that there are going to be pros and cons to all of these models. One of the biggest cons to this model is oversimplification. This is a very simple cartoon of a three-dimensional, very complex shape. Please remember that while we use this kind of a shape, it's not exactly the way they look. After the chemical reaction has occurred, it will produce products, which it will release. Now it's free. It can take another substrate. It's a reusable source. It can be reused many, many times. And when they're no longer needed, they can be broken down and reused, allow their components to be reused inside the cell. Things to remember, just a little bit of additional. Proteins can be broken down or altered in shape permanently by things like heat, so excessive heat or excessive loss of heat. It's too cold, the enzyme doesn't function. pH, if it's outside its normal, it won't work well. It could possibly bend it completely out of shape where it never works again. And then of course salt, too much, too little can affect whether or not it can function. So things to be aware of. All right, last thing on the chart are our nucleic acids. So these are going to have CHONP. They're going to be made of the monomer, which is called a nucleotide. It's composed of a phosphate group, all of them are the same, a 5-carbon sugar, which is either deoxyribose in cases like DNA, or ribose in cases like RNA and ATP, ADP, GDP, GTP, any of those, and nitrogenous base. The base is the major issue. When we're thinking about how they differ, there are 4 bases. The order of the bases, just like order of amino acids, is going to be significant, and it's going to change the way these things work. When we're talking about their polymers, the polymer of DNA is double-stranded. A is bonded to T, C is bonded to G. They have double helix structure, and they use deoxyribose. RNA is another polymer, and it's going to be single-stranded. The A will bond to U instead of T. C and G still bond together, and it's going to be made using the sugar ribose. ATP is the other one that we really think about, and that is an adenine combined with a ribose sugar and 3 phosphate groups. We use this for cellular energy. The last phosphate group is popped off to release energy or put on to store the energy. If we're looking at 3 phosphates, it's ATP for triphosphate, and if we only have 2, it's ADP for diphosphate. The function is going to be to code for protein synthesis. That is the actual code that puts the amino acids in the correct order, or at least that's what it's supposed to do. This is the inherited particle of genetics that Mendel discussed in his work. And then ATP and ADP, and along with GDP and GTP, although we don't use those very often, are for cellular energy. So just some basic things to be aware of. The last thing I wanted just to kind of quickly review with y'all is our dehydration, synthesis, and hydrolysis. So the way that we make polymers and the way we break polymers down back into their original monomers. So dehydration, synthesis, when we think about the word, to dehydrate is to remove water, to synthesize. Synth means to make. And so we have a basic biomolecule monomer, a basic biomolecule monomer, and all of them will have some sort of an H and some sort of an OH. They will be facing each other. To combine them, we take the H from one and the OH group from the other. That is released as water, and then we can have a bond formed between them. And that is going to form some sort of a covalent style bond and a water molecule. So as we create polymers, we release water. Hydrolysis is the exact opposite. So hydro meaning water, and lysis meaning to break down. We have a polymer of some sort, and we have to add water to it. That is going to provide the hydrogen and the OH necessary for each of those monomers to split back into their original shape. So we release water when we build polymers, and we need water when we break polymers. So as far as like getting that straight in your head, I want you to think about it like this, okay? When a small child or baby is growing, they're having to build a lot of polymers really, really quickly. And little children and babies, as you are probably aware, have to pee all the time. Part of it is that they don't have bladder control, but another part of it is they are making so many polymers so very quickly that they have to get rid of all that excess water, hence the need to pee. When we're breaking down polymers into monomers, this is why it's so important to have, one of the reasons, so important to have water in our daily diet, and it's one of the major reasons that water is required for your intestines to work well, because that's one of the things that's used to break those things down. And so a lot of cultures will drink water at meals, or if you're trying to lose weight, they tell you to drink a glass of water with your meals, and that's to help the food break down. Okay? So, as always, if you have questions, send me a text or an email. I'm happy to take care of it. These posters and the other ones, I'm going to take photographs and load those up for you so that you have those that you can review a little bit closer, because I know it's hard to see them as we're going through. Know that I am confident in your ability. I know that you have prepared yourselves for this exam. Just take a deep breath and know that I believe in you, and I am proud of the effort that you've made all year long. Okay? Mwah. Be well.